GIT, whatever we have discussed in the last class, okay, it will be followed. Last week we have discussed about the stomach and esophagus and stomach. Esophagus and stomach both are the part of GIT. Okay, now after esophagus and stomach, what is the next part? Small intestine and large intestine. Okay, so small intestine, large intestine, both they are the part of GIT. So how many layers will be there? Four basic layers are there. Please by heart those layers. What are the name of those layers? Mucosa, submucosa, muscularis externa, serosa or adventitia. Okay, those four basic layers. It will be present over here also. But there will be some modifications or there will be some changes. Okay. So in case of small intestine, first we will see the small intestine, then we will go to the large intestine. So you know that the main function of the small intestine is to complete the digestion and start the absorption. That is the main function of small intestine. Now in case of small intestine, in order to do the absorption, okay, some modifications are there. For example, extensive length is present. If you see the length, it is approximately 6 meter, the length of the small intestine. Okay. Next, there are circular folds present. That is, if you see the interior of the small intestine, circular folds are there. And what are the name of those folds? These are called as plicae circularis. Okay, these are called as plicae circularis, or also called as valves of Kerkring. And these are permanent folds. Okay. In case of stomach, you have seen gastric rugae are there, but those gastric rugae, these are temporary. Here, these are permanent in nature. Okay, which can be seen easily. Now, why this particular fold is permanent in nature, these circular folds? Because all the layers of the mucosa is extending into that fold, and sometimes the submucosa is also extending into the fold. Because of that reason, the circular folds are continuous in nature. Okay, what is the function of that fold? It slows down. Okay, whenever the food is traveling through the small intestine, it will slow down the speed, so that the time will be more for absorption. Next, another modification which is present inside the small intestine, that is villi. Okay, last week I have shown you the structure of villus. What is present inside the villus? What is the function? Already we have discussed. So that is a modification, okay, which is present in the lumen of small intestine. Okay, that means if you go to the stomach, villi are not there. If you go to large intestine, villi are not there. Villi are absent. Only the villi are present in case of small intestine. So that is a important modification. Okay, so that will also facilitate the absorption. Okay, now if you see the villi, they are present in duodenum also, they are present in jejunum, and they are present in ileum. All three parts villi are present, but there is a difference. What is the difference? The size of the villi is not same, shape of the villi is not same, and the number of the villi per square centimeter is different. So these are some changes of the villi. Remaining structure, function same. Okay, number, shape will be different. Okay, and the size will also be different. So in the first part of the duodenum, if you see, they appear as broad ridges. Okay, become tall and foliate in the distal duodenum. So see, first it will be broad ridge like, then it will become tall foliate in the distal part of the duodenum and proximal part of the ileum, jejunum. Now gradually it becomes shorter to form finger like in the distal jejunum and ileum. So like this, the transition is taking place in the shape of the villi. Okay, as you are going from duodenum to jejunum. Next, villi, the density also changes. See, 10 to 40 per square millimeter. That is the number of villi, and it varies. Okay, in case of duodenum it is more, in case of jejunum it is less, and in case of ileum it is very less. Okay. Next, here you can see the height 0.5 to 1 millimeter in height. That means it is not microscopic. The villi are not microscopic. Okay, you can see the villi with the help of magnifying lens. Okay, with the help of magnifying lens, villi can be seen easily. These are not microscopic structure. Next, another modification is there in the lining epithelium. Now, what is the lining epithelium of small intestine? Last week we have discussed about the lining epithelium of GIT, simple columnar epithelium. Simple columnar epithelium. So here also it is simple columnar epithelium, but there is a modification. And what is that modification? Microvilli are present. Okay, on their surface microvilli are present. Now that also striated border. Now can anyone tell me what is striated border microvilli? If you see the shape of the microvilli, दो type का होता है. One is striated border, another one is brush border. In case of striated border, okay, the microvilli are equally placed. One microvilli, another microvilli, they are equally placed. They are parallel to each other, and the shape and size of the microvilli are also same. Okay, as a result, they will appear a straight line over the surface of the surface epithelium. That is called a striated border. And opposite होता है brush border. That means the dimension of the microvilli are not same and they are not parallel to each other. That's why it is called as brush border. Okay, so two types are there. Next, crypts of Lieberkühn. This is nothing but the pits which you have studied in case of stomach. And deep to the pits we have seen gastric glands are present, if you remember, right? So similarly, in case of intestine, those depressions are called as crypts, and deep to the crypts intestinal glands are present, and they are also called as crypts of Lieberkühn. Okay, so these are simple tubular intestinal glands extend into the lamina propria up to the muscularis mucosa. Okay, but here the glands which are present over here, simple tubular gland, and the glands which we have studied in case of stomach, okay, some differences are there, especially in the cells. The cells which are present in this particular glands, these are different from the cells which are present in case of stomach. Okay, now these glands are lined by tall columnar epithelium that is continuous with the epithelial lining of the villi. Now, two diagrams are here. Okay, now in this diagram you can see this is a common structure for small intestine. Now this is the lumen of the small intestine. So in the lumen of the small intestine you can see these finger like projections from the lumen of the small intestine, and these are called as villi. Okay, now in between the villi, the spaces which are there, these are called as intervillous space. Okay, next if you see the intervillous space, can you see the depressions? Can you see the openings? These are called as opening of the crypts. Okay, and you can see from the crypts it is going downwards, and in this particular area what will be present? Intestinal glands. Okay, are also called as crypts of Lieberkühn. Appearance is almost similar like the stomach. Only difference is here we are getting the villi. In case of stomach we have not seen the villi. Okay, next other layers, can you see it is almost same. Okay, so here you can see the lamina propria. ये देखो पूरा lamina propria. Here also you can see the lamina propria. Then here you can see muscularis mucosae. You can see muscularis mucosae. Then here submucosa. Okay, here you can see the submucosa, and below submucosa muscularis externa is present. Okay, and then the serosa. Now some part it will be serosa, some part it will be adventitia, depending whether peritoneum is present or absent. Okay. In case of small intestine, okay, in case of small intestine, especially in duodenum, if you see the submucosa, the speciality of the duodenum is submucosal glands are present, and these are also called as Brunner's gland. Mucus secreting glands are present, and these are called as Brunner's gland. So the submucosa is packed with Brunner's gland in case of duodenum, and that is the most important unique identifying feature of duodenum. Okay, because see, in case of small intestine, three parts are there, right? Duodenum is there, jejunum is there, and ileum is there. Now microscopically how we will differentiate? क्योंकि चारों लेयर है, म्यूकोजा, सब म्यूकोजा, all those layers will be there. तो कैसे आइडेंटिफाई करोगे? How will identify? And more or less they are same. So the best way is focus on the submucosa. If you focus on the submucosa of duodenum, what you will see? Submucosal glands. Okay, mucus secreting submucosal glands. These are also called as Brunner's gland. That is only present in case of duodenum. In other, these are absent. So like this, you can easily differentiate between the duodenum and other two parts. Okay. Now this already you know, we have already discussed about the interior of small intestine. Okay, so here you can see the plica circularis, or also called as the valve of Kerkrings. Okay, now we have to go one by one, layer wise. So in mucosa, what is the first layer? Now you know that mucosa is made up of three distinct layers, right? Mucosa is made up of three distinct layers. What are they? Yes. How many layers are there in the mucosa? Three layers. What are they? First is lining epithelium, lamina propria, and muscularis mucosae. Okay, that means first is the lining epithelium. So what is the lining epithelium in this case? Simple columnar epithelium with microvilli. In bracket you will be writing striated border. Completely, complete sentence. ठीक है? Don't write only simple columnar epithelium. No. Stomach में simple columnar epithelium रहता है, that also mucus secreting. Here we are getting simple columnar epithelium, that also striated border. And for secretion of the mucus separately, we are getting goblet cells over here. Okay, that you have to mention. So intestine is lined by simple columnar epithelium. After that, what you have to write? In a bracket, you have to write with microvilli, having microvilli and striated border. Okay. Next, different types of cells are present in the mucosa. Different types of cells are present in the small intestine. Okay. What are those cells? So you can see enterocyte. Now, please note, keep that in mind. जो lining epithelium है, okay, the lining epithelium in case of small intestine are also called as enterocytes. Okay, enterocyte. The other name of the lining epithelium in case of small intestine is called as enterocytes. Okay, so don't get confused. Enterocyte means lining epithelium. So here you can see tall columnar cell, basal oval nucleus, and striated border. Okay, this is the most important identifying feature. Next, what is the function? Absorption, secretion of some enzymes, etc. Next, goblet cells are there. Okay, so along with the enterocytes or the lining epithelium, we are getting goblet cells. Okay, so goblet cells, why they are kept? Why they are present? To secrete mucus. Okay, very important. So here also similar appearance will be there. That means in the goblet cell you will see it is empty whenever you will see under microscope, because whenever the processing was done, the mucus which is present, it is completely removed, washed away. Okay, in order to see that particular mucus, what you have to do, a special type of stain is used, that is called PAS, periodic acid skiff method, in which it will appear magenta color. Unicellular and mucus secreting. This is the function. Next, these are some important cells. Okay, these are some important cells which are present. Okay, now these particular cells, they are mainly present. Okay, these particular cells, they are mainly present in the intestinal glands or crypts of Liverpool. Okay, if you see the enterocyte goblet cell, okay, they are placed together in the lining epithelium. Next, as I said, the crypts are there, or intestinal glands, crypts of Liverpool. In that particular gland, these different types of cells are present. Okay, so first, Paneth cell, or also called as zymozyme cell. Okay, highly acidophilic cytoplasm, oval basal nucleus, zymozyme granules are present at the apical part of the cell. Okay, what is the function? Phagocytosis. Okay, secretion of lysozyme alpha defensin. Okay, and with the help of this, they maintains the normal bacterial flora which is present in the small intestine, and that also helps in digestion. Next, enteroendocrine cell. Okay, these are also called as argentaffin cell. Why? Because they has to be seen. They can be seen with the help of silver salt only. Okay, that means if you want to stain the cell, enteroendocrine cell, which type of stain is required? Silver salt. That's why they are called as argentaffin cell. Okay. तो इसका फंक्शन क्या है? दे सिक्रेट डिफरेंट टाइप्स ऑफ पॉलीपेप्टाइड्स, फॉर एग्जांपल एनकेफेलिन्स, वैसोएक्टिव इनहिबिटरी पॉलीपेप्टाइड्स एक्सेट्रा. ओके, तो इसका मेन फंक्शन क्या है? दिस एंटेरोएंडोक्राइन सेल, they will control the motility of the intestine, they will control the secretion, they will control the absorption, blood flow, all those things are controlled by these enteroendocrine cells. Okay, next, M cells are there. This is very important, present in the epithelial lining, okay, over the Peyer's patches. Now iska function kya hai? These are antigen transporting cell. That means they will transport the antigen to the lymphocytes and dendritic cell so that the antibody can be produced. कोई भी एंटीजन आता है इन द स्मॉल इंटेस्टाइन, दिस पर्टिकुलर सेल विल कैरी द एंटीजन, उसमें एक तरफ का रिसेप्टर्स रहता है, so that will bind to the antigen and it will be produced in front of the cells which can produce antibodies against it. Okay, so that is the function of the M cells. Next, stem cells are there, so already you know what are the functions of stem cells. Now this is the diagram. Okay, so please keep that in mind. This particular chart is important, which you have to write in your practical notebook. Okay, and along with that you have to draw this particular diagram also, right? In this diagram you can see this is the enterocyte cell, tall columnar cell. You can see the microvilli giving striated appearance. Okay, striated border appearance. Next, here you can see this is the goblet cell. If you see the shape of the goblet cell, it is mostly wine glass appearance. Okay, and here you can see the secretory vesicles are present. Okay, then you can see the enteroendocrine cell and you can see the Paneth cells also. So this diagram is also important. Don't forget to draw it in your practical notebook. So after lining epithelium, what is the next layer? It is the lamina propria. तो लेमिना प्रोप्रिया में क्या रहता है? It is made up of reticular fibers. Okay, along with that lymphatic aggregations are present. Now this is very important in case of small intestine. Okay, the lamina propria, aggregation of lymphatic populations are there. Okay, plasma cells and other components of the immune system are also present. Next, solitary and aggregated lymphatic follicles are present in the lamina propria. Okay, they are forming the pairs patches and they constitute the GALT. What is meaning of GALT? Gut associated lymphoid tissue. Okay, so this solitary and aggregated lymphatic follicle together, they are forming pairs patches and it comes under GALT, that is gut associated lymphoid tissue. Okay. Now in case of small intestine, ये जो pairs patches है, okay. Now these pairs patches you will see in case of duodenum they are very less. Generally it is not seen. But if you go to jejunum, ileum, the number of pairs patches are more. Okay, in jejunum it is less. In case of ileum it is more. That means whenever you will focus on the lamina propria of the ileum, at that time you can see this collection or aggregation of the lymphatic follicles. These are nothing but the pairs patches. Very important. Okay, so as you can see, they are most prominent in the terminal ileum. Now their lymphoid tissue may be extend into the submucosa. Okay, so from lamina propria, the amount of the lymphatic tissue is huge that it is extending into the submucosa, especially in case of ileum. So that is also an important identifying feature. Now for duodenum, already I have told you how you will identify the duodenum. Three parts are there of the small intestine. How will identify the duodenum? कैसे identify करोगे? You will see the submucosa. In submucosa, we will get submucosal glands, okay, or also called as Brunner's gland, which is absent in case of jejunum and ileum. ठीक है? Next, तो duodenum हो गया, अभी jejunum ileum है. How will identify? You will concentrate on the lamina propria of the jejunum and ileum. In case of jejunum, hardly you will see lymphatic follicles. In case of ileum, lot or plenty of lymphatic follicles can be seen. Okay, and those are called as pairs patches. So like this you can identify, and the amount is so huge that sometimes it is extending into the submucosa. Like this we can identify the jejunum and ileum. Other identifying features are also there. As we will go further, you will come to know. Okay, next is the muscularis mucosae. Now what is present in case of muscularis mucosae? Muscularis mucosae is same in case of same like the stomach. No changes are there. Okay, that means outer longitudinal layer and inner circular layer and outer longitudinal smooth muscle cells are present. Okay. Now the few fibers extend into the mucosa and help in the movement of the villi and the expulsion of the secretion from the intestinal glands. Okay. Next, in the submucosa, it is made up of loose connective tissue. Okay. It is made up of loose connective tissue where lymphatics, nerves are present, blood vessels are present. Okay. Now, in case of submucosa, reached elevation from the coats of circular folds are present, that is the collagen fibers, elastin fibers, okay, in a considerable amount. These are also present in case of submucosa. Now this provide adequate support, elasticity and strength during the peristalsis. Okay, so in case of submucosa, the fibers which are present, okay, the collagen fibers, elastin fibers, during the time of movement, that is peristalsis, they are providing support, okay, they are providing elasticity and strength to the small intestine. Okay, next, in case of submucosa, as I said, Brunner's gland are there, which is the most important feature to identify the duodenum. Okay, now if you see the Brunner's gland, these are branched tubular gland secreting mucus, which is rich in bicarbonate ion. तो इसका भी function है to secrete mucus, but here the mucus is rich in bicarbonate. Okay, if you see the pH, it is I think more than 8 or it is approximately 9, the pH of the bicarbonate ions which are secreted by the Brunner's gland. So first Brunner's gland जो होता है, what is the main function? What it will do? It will produce mucus, and in this mucus it is rich in bicarbonate. So what is the function? Yes, because the medium of the small intestine is alkaline in nature. Okay, if pH is 9 or more than 8.5, what does it indicate? It is alkaline. So क्यों alkaline होता है? Why? Because from the stomach, whatever it is coming to the duodenum, it is acidic in nature. So that acid has to be neutralized. For that, it is rich in bicarbonate. Okay. If you have studied succus entericus in physiology, you can correlate. Okay. If you have studied, just correlate, you will understand what is succus entericus, what is the function, what are the constituent of succus entericus. Okay. Muscularis externa. Now in case of muscularis externa, again two layers are there, that is inner circular, outer longitudinal, okay, where the myenteric plexuses are present, okay, which helps in local mixing and as well as peristalsis. And lastly the serosa. Now in case of serosa, most of the small intestine is covered by peritoneum. Okay, most of the small intestine covered by peritoneum, that is the mesentery. Some part which is not covered, there you will be getting the adventitia. Best example is duodenum. We know that duodenum mostly it is retroperitoneal organ. Okay, so whatever we have discussed till now, that was common for entire small intestine. Now specifically if we have to study the duodenum, okay, then what exactly you have to note. So in case of duodenum, first of all these layers are present. Okay, now here you can see the villi. So already I have mentioned, in case of duodenum, if you see the villi, please keep that in mind, they are taller as compared to other two parts. Number of villi are more as compared to other two parts. Okay, and along with that, in the submucosa, you will be getting Brunner's gland, or also called as mucus secreting gland. Okay, other things are same, no change. Okay, so here you can see in case of duodenum, mucosa, tall columnar epithelium, striated border. Okay, it is same, finger-like villi, crypts of liver cone in the lamina propria, muscularis mucosae. Now here you won't be getting any pairs patches. Okay, here you won't get any pairs patches. Submucosa, see, very important. Brunner's gland, highly branched tubuloalveolar gland, mucus secreting gland, rich in bicarbonate. Muscularis externa, two layers, inner circular, outer longitudinal. And adventitia or serosa, because the duodenum is retroperitoneal organ. Mostly it is not covered by peritoneum. In that part you will be getting the adventitia. Now here is the diagram. Okay. Now here you can see the villi. Okay, you can see these are the villi. See, this is one villi, this is one villus, this is one villus, this is one villus. In between them, can you see the space? These are the intervillar space. Ye dekho, these are the intervillar space. Okay. Next, this is the lining epithelium. See, this is the lining epithelium. This is the lining epithelium. Okay. Now in the lining epithelium, can you see some empty spaces? Can you see? So what are these empty spaces? These are goblet cells. Okay. Empty spaces जो appear होगा goblet cells से. ठीक है. Next, see clearly you can see the striated border also, but generally in low magnification ये दिखाई नहीं देता है striated border. Okay. In high magnification it can be seen. Next, so what are these cut sections? This is the lamina propria, right? This is the lamina propria, this is the lamina propria. This cut section, these are nothing but the intestinal glands, or also called as crypts of Liverpool. Okay, just now we have studied the cells which are present. Okay, so I was talking about these cells, these glands. Okay, deep to that you can see this is the muscularis mucosae. ये देखो, this is the muscularis mucosae. Okay, next layer, this is the submucosa. Can you see the glands? In submucosa you can see the Brunner's gland. See, this is the Brunner's gland. Okay, now please keep that in mind. ये देखो, I want to show you difference, means how it will be easier for you to identify. Now this is also a gland, these are also glands, these are also gland. Can you see the difference? Huh? No, not like this. See the staining characteristic. See why these are empty, totally empty. Why these are totally empty? जो Brunner's gland होता है, okay, if you stain them with the help of H&E, mostly it will become empty. Why? Because mucus secreting gland है. Mucus are completely removed. But these glands, these are not mucus secreting gland. For mucus secreting already we are getting the goblet cell. They will secrete the mucus. That's why they are empty. Here these are not secreting mucus. Okay. So this is how you can differentiate between the glands. तो ये Brunner's gland हो गया. If you go below, then which layer is this? Muscularis externa. In muscularis externa you can see two distinct layers followed by the serosa. Okay, so this diagram is for which part of the small intestine? Duodenum. Okay, this you have to draw, this one. Okay, so this is under low magnification and this is under high magnification. Okay, now here you can see the villi. ये देखो villi है, this is one villi, this is one villus, this is one villus. These are the intervillus space. Okay, now if you track down the intervillus space, okay, you can see these are the glands, intestinal glands. See, these are the intestinal glands. Okay, next this particular part, this is the muscularis mucosae. Okay, this is the muscularis mucosae. Okay, next below that, what is this? Submucosa. In submucosa, can you see the Brunner's gland? Plenty of Brunner's gland can be seen in the submucosa. You can see plenty of Brunner's gland. Means the submucosa is packed with Brunner's gland. Easily it can be seen. Okay. Next, if you go below, then this entire part is the submucosa. Then this is the muscularis externa. Two distinct layers can be seen easily, followed by the serosa. Okay. Now this is under high magnification. This you don't have to draw. Just draw this you have to draw this one. This one. This is for your understanding, where you can see this is for the low magnification and this is under high magnification. Okay. In high magnification, you can see what is this? Anyone? ये क्या है? One villi. See, villi means multiple. Villus means singular. एक है तो villus. That means it is a single. That's why it is called as villus. Okay. Now, how the lining epithelium and villi are oriented? That day I have shown you. I don't know you remember or not. In case of small intestine, if you see the lumen of the small intestine, okay, if you concentrate over here, okay, so this is one villus, okay, this is one villus. Now on the villus, how the lining epithelium is oriented, okay. So as you can see over here, first one villus is there. Inside the villus, as I have mentioned earlier, one lacteal will be there centrally placed, okay. Then capillary plexus is capillary bed is there, which is having an arterial end and having a venous end. Okay. Next, on the surface of the outer aspect of the villus, okay, the central core is made up of connective tissue. Central core is made up of connective tissue. On the outer aspect, we are getting the lining epithelium, and over the lining epithelium, we are getting microvilli. So this is the orientation of the villi and microvilli along with the lining epithelium. ठीक है, तो पहले जो विलस है, जो लुमेन है, लुमेन से फिंगर लाइक प्रोजेक्शन निकलेगा, दैट इज द विलस, आई एम टॉकिंग अबाउट वन. ठीक है, इसका जो सेंट्रल कोर है उसमें कनेक्टिव टिश्यू रहता है. कनेक्टिव टिश्यू में क्या रहेगा, सेंट्रली प्लेस लेक्टियल रहेगा, ओके, देन आर्टेरियल साइड रहेगा, विनस साइड रहेगा एंड कैपिलरी बेड रहेगा. ठीक है, देन on the outer aspect we are getting the lining epithelium, which is simple columnar epithelium. Okay, along with that goblet cells are also there, and on that surface of that lining epithelium we are getting the microvilli, that also striated border. This is how it is oriented. Okay, that you can see over here. I know by looking at this diagram you will not understand, that's why I have drawn it separately. Okay, next after duodenum, what is the next two part? Jejunum and ileum. Okay, just see the differences between jejunum and ileum. क्या रहता है? What is the difference? In case of jejunum, you can see lining epithelium is same. Okay, but here, as I said, if you see the villus, number of villi will be less, and the shape of the villi will also change over here. In case of jejunum, if you compare with it, the duodenum, and the length of the villi will also be less. Next, here crypts of Libercun in the lamina propria, muscularis, mucosa, and here you won't find any of the Peyer's patches. If it is there, that also it is not significant, very less quantity. Okay, in the lamina propria. Submucosa, connective tissue, no Brunner's gland are there, and Peyer's patches. Okay, nahi rehta hai. Okay, in some cases I have seen it is present, but mostly it is absent. Next, muscularis externa, same. Okay, but here you will be getting completely serosa. Okay, no adventitia, because it is completely covered by peritoneum, intraperitoneal organ. Next, in case of ileum, same, everything is same. Okay, the difference is in case of villi, so only few villi will be there, and the length of the villi will be shorter as compared to the jejunum. Okay, Brunner's glands are absent. Now, lamina propria consisting of intestinal glands and Peyer's patches. Huge amount of Peyer's patches are present. Okay, in lamina propria, and sometimes it is extending up to the submucosa. So that is the difference between the jejunum and ileum. Now here you can see this is the histology of jejunum. Okay, so what you can see, so what are this? These are the villus. This is one villus, this is one villus, this is one villus. Okay, so same orientation whatever we have discussed, but here you can see less number of villi are present. Okay, then here glands are present. Okay, but some amount of lymphatic nodules can be seen in the lamina propria. In jejunum it is less, but it is present. Okay, next here you can see submucosa, इसमें कोई भी gland नहीं है, no Brunner's gland or no submucosal glands, mucus secreting glands are there. Other layers are same. Okay, so this diagram you have to draw for jejunum. In histology of duodenum, 3 diagrams you have to draw: duodenum, jejunum and ileum separately. Okay, see the diagram carefully. So see, these are the villi. ये देखो, these are the villi. Okay, next these are the intervillar space. After that, these are the glands. Okay, see, these are the glands. After that, here you can see the submucosa. Okay, in submucosa you can't see any of the gland. Okay, then these are the muscularis externa, 2 layers, and then this is the serosa. Now what is this? This is the histology of ileum. What you have noticed over here? First, see the villi. The length of the villi, these are shorter. See, the length of the villi are short as compared to the duodenum and jejunum. Next, focus on the lamina propria. Focus on the lamina propria. ये देखो, focus on the lamina. What you can see? Peyer's patches are more. Okay. Previously we have seen almost it is absent, but here bundles of Peyer's patches, or lot of Peyer's patches are present. It means more aggregation of lymphatic follicles are present. Okay, So like this you can easily differentiate, and see some are extending into the submucosa also. And here also in submucosa no glands are present, Brunner's gland nahi rehta hai. Okay, and this particular part, this is the externa, muscularis externa, followed by the serosa. Okay, now this is the schematic diagram which you have to draw, and this is the original diagram of histological diagram of ileum. Okay, so that is all about the that is all about the small intestine. Okay, now we'll discuss about the large intestine. So I hope you have seen the large intestine today, everyone. What you have seen in large intestine? All the parts you have seen. Have you seen the cecum? Have you seen the appendix? Okay, very good. Now, can anyone tell me what are the three important external identifying features of large intestine? Yes, appendices epiploicae, tinea coli, and hostration or circulation. So, what are appendices epiploicae? These are fat-filled peritoneal sac which are attached to externally to the large intestine. Okay, as you can see over here, these yellow color things. Okay, these are the appendices epiploicae. Okay. Next, what is tinea coli? What is tinea coli? How it is formed? See here you can see the tinea coli. ये देखो tinea coli दिखाई देता है. Okay, I hope you have seen the tinea coli, right? तो जो longitudinal muscle fibers होता है, we know it is muscularis externa में दो layers होता है, inner circular and outer longitudinal. So that outer longitudinal layer is modified in case of large intestine to form the tinea coli, and because of that tinea coli, okay, it is pulling the small intestine because the circular folds are longer as compared to the longitudinal fold. That's why the hostrations or seculations appear. जो hostration seculations होता है in case of large intestine क्यों होता है? Why? Do you know that hostration seculations are present? क्यों होता है? Why? Because of this tinea coli. Okay, if you see the length of the tinea coli, it is shorter. And how the tinea coli is formed? It is the modification of the longitudinal muscle fibers, bundles of the longitudinal muscle fiber. So just circular one hai, it is longer. Okay, so what it will do? It will pull the wall of the intestine. So due to that pull, these hostrations or seculations are produced, which you can see. So this is the external features of the large intestine. Okay, already you have studied, you have seen. That is the seculations or hostrations, tinea coli, and appendices epiploicae. And here you can see all the parts of the large intestine. What about this? This is the internal structure of large intestine. Okay. Now, if you see the internal structure of large intestine, same all those four layers will be there. Okay. But what will be the difference? If you see the lumen, so this is the lumen. Okay. What is absent over here? Villi are absent. That means you can't see any villi. Rest everything is almost same. Okay. So lining epithelium will be same. Then you can see opening of the crypts. Crypts, it will go down to form the crypts. This one, glands, intestinal glands. Okay. But here in intestinal glands, the M cell, Penneth cell, all those cells are not present. Okay. Only mucus secreting cells are there over here. Okay. Next, you can see other layers, muscularis mucosa, okay, then submucosa, below that muscularis externa, okay, serosa, all those things will be present as usual. Okay, so in histology of large intestine, we'll be considering the colon. Okay, if you take the example of colon, so all the layers are present, mucosa, submucosa, externa, and serosa. Okay, now some part it will be serosa, some part it will be adventitia. So in case of mucosa, it is lined by same simple columnar epithelium which is absorptive in nature, where it is having microvilli, and along with that goblet cells are there. Same like the small intestine. Next, lamina propria filled with intestinal glands are also called as crypts of Lieberkühn. These glands are lined by goblet cells, mainly the mucus secreting cells. Okay, very simple. Okay, lamina propria shows discrete lymphatic follicles. Now lymphatic follicles are present, but the numbers are very less. Okay. Submucosa consists of dense irregular connective tissue. Muscularis externa, same inner circular, outer longitudinal. But here a modification is there. Now the longitudinal fibers, they form 3 bands of muscle fiber. These are called as tinea coli. Okay, this you have to write in the practical notebook, and during the time of exam also, if it comes, you have to mention the modification of the tinea coli, modification of the longitudinal muscle fibers which is forming the tinea coli. Okay. Lastly, you can see colon doesn't have plica circularis. So if you see the internal structure, the circular folds, those are absent, villi are absent, and pennate cells, as I have mentioned, these are also absent. Okay. Now here you can see what are present in the different layers of the colon. Okay. Now this is the diagram. Okay. Now in this diagram, what you can see, so this is the lumen of the large intestine. Okay. Now here you can see this is the lining epithelium. This is the lining epithelium. Okay. Now lining epithelium is simple columnar epithelium, and this goblet cell, whatever you can see the empty spaces, these are nothing but the goblet cells. Okay. Next here you can see this is the lamina propria. Here देखो lamina propria. Now in lamina propria, as I said, some lymphatic follicles are there, very few. Okay. Next, this is the muscularis mucosae. Here देखो, this is the muscularis mucosae. Okay. Next, you can see the intestinal glands also. See, this is the cut section of intestinal glands. Now here, if you see the intestinal glands, so many empty spaces you can see. Okay. In the intestinal glands, you can see the empty spaces. Why you can see empty spaces? Because as I said, these glands are mucus secreting gland. Previously, we have not seen empty spaces in case of small intestine, if you remember. But here you can see. Why? Because these are nothing but the mucus secreting cells. That's why they are appearing empty. Next, this is the submucosa. Okay, made up of dense irregular connective tissue. Okay, no Brunner's gland, nothing is present over here. Okay, then this particular layer, muscularis externa, 2 layers. The outer layer is modified to form tinea coli. Tinea coli generally microscopically दिखाई नहीं देता है, because that you can see directly on the wall of the large intestine. So that cannot be seen microscopically, but you have to mention. Okay. Now, so this is the schematic diagram which you have to draw. This is for large intestine. Okay. And after that, in a bracket, you have to write colon. Okay. Next, this particular image, histological image of large intestine, this is under low magnification and this is under high magnification. Okay. Now, if you see the histological image of small intestine and large intestine, have you noticed one thing? Small intestine, 3 parts already have shown you. Now you are looking at the large intestine, histological image of large intestine. Have you noticed any difference? I know Brunner's glands are not there, Peyer's patches are not there, villi are absent, all those things we have discussed. One thing what I have not mentioned, can you notice that? Okay, so the most important part is, if you see the slide under microscope, especially of the large intestine, more number of goblet cells are present. Okay, more number of goblet, that's why you will see plenty of empty spaces are there. More number of goblet cells, which is less in case of a small intestine. That is also important identifying feature in case of large intestine. Identification of large intestine, small intestine उतना difficult नहीं है. Okay, हो जाता है. Okay. But I have seen students used to commit mistake when they will see the lumen, they think that villi is present, directly they will write it as small intestine. Okay. So that you have to understand. That's why what you have to do, you have to study about the structure of the villus. You have to know how the appearance of the villus is taking place under a microscope. Okay, that you have to correlate and then study, or else you cannot identify directly the small intestine and large intestine. You will get confused. Okay. In case of large intestine, if you remember, there is a part that is called appendix. In large intestine, appendix is also there. Okay. You have seen the appendix. You have studied about the appendix, parts of the appendix. Okay. Then different positions of the appendix. Right. 11 o'clock, 12 o'clock position. Okay. Now, so appendix is also a part of large intestine. That means all the layers of the small intestine will be present in the appendix. Okay, everything is present. Now, what additional things are present in the appendix? That means if you get a slide of appendix and if you get a slide of large intestine, how will differentiate? In case of a large intestine, already you have seen this is the slide of large intestine. Okay. Now, in case of appendix, what you can expect? What will be the difference? Not lymph nodes. Lymph node cannot present. Lymphatic tissue can present. Lymphatic nodules can present. Okay, so in case of appendix, see all the layers are present, same lining epithelium, lamina propria, okay, same. But here the difference is, if you see the lamina propria, numerous lymphatic follicles are there, okay, and these lymphatic follicles, they extend into the submucosa. Lots of new lymphatic follicles are there. Okay, like this you can easily identify or you can differentiate between the large intestine, colon, and this one appendix. Other layers are same. And another important difference, can you tell me the difference, especially in case of appendix? Now I have mentioned that in case of large intestine, what you are getting three important identifying feature, that is appendices epiploicae, tinea coli, horstations and speculation. Now these are extending up to sigmoid colon. Beyond that, these are not present, if you have noticed. Okay, and this tinea coli is absent in case of appendix. tinea coli appendix में नहीं रहता है. Okay. Okay, so here you can see mucosa, submucosa, muscularis externa, where tinea coli is absent. Tinea coli नहीं रहता है. Okay, so that thing also you have to mention. Now here you can see histology of appendix, the schematic diagram. Okay, so you can see this is the lumen of the appendix. Now this is simple columnar epithelium. Okay, and along with that you can see the intestinal glands. So these are nothing but the mucus secreting glands. That's why you can see these are completely empty. Next over here you can see the lymphatic nodules. Okay, lymphatic follicles, aggregation of lymphatic follicles which are present, and sometimes you can see these are extending up to the submucosa also. Okay, so this is the important identifying feature in case of appendix. Okay, like this you can differentiate between the appendix and the large intestine. Okay, next this is the submucosa, then this is the muscularis externa. In muscularis externa, and after that serosa. In muscularis externa, please mention that tinea coli are absent. Okay. Now what about appendix, whether it is intraperitoneal or retroperitoneal? Which type of organ is this? It is intraperitoneal, completely covered by peritoneum. What is the name of the peritoneal fold? Mesoappendix. Okay, so that is all about the large intestine.